Hello everyone, and welcome to the third and final part of lecture number 2 here. We're still moving along in the endocrine system, which happens to be Pearson's chapter 18 here, and we're continuing with the hypothalamus and the pituitary gland as we've covered in this lecture. So we're getting through the final details here. I'm going to talk about how the hypothalamus is linked to the pituitary gland, talking about something called the hypophyseal portal system. And then here you can see histological images, and I've been stressing pretty early in the semester, and if you've had me in the previous semester, you know that I've stressed perspective, and so histology is a very unique perspective. We are looking at, essentially the tissue and cellular level, and so you need to know a few things about this. And then finally, we'll wrap up with kind of the role of growth hormone. Obviously, growth hormone is a pituitary hormone, but we're going to start talking about its impacts when things go awry with growth hormones. We're going to start introducing you to what happens when hormones aren't secreted appropriately or are kind of secreted at a too high of a level or too low. So let's get into this. As usual, I have my objectives. If you want to read over them, these were from the first part, these were from the second, and this is what we are covering today. So if you look at parts 1 and 2 and are confused by those objectives here, maybe you want to listen to the part 1 or part 2 of lecture number 2. Otherwise, if you're like, yeah, I remember these and feel pretty good about some of those concepts, at least feeling like, yeah, I remember the terminology here, certainly moving on to part 3 is a perfect timing. So let's get into the lecture here. So we start off with something called the hypophyseal portal system, and this image at this point should be really similar. I've used it in all 3 parts here, but if you notice, we have nuclei attached here. So here's supraoptic nuclei, here we have paraventricular, here we have neurosecretory neurons. That one I'm not too concerned with you getting caught up on, but we've already talked supraoptic and paraventricular, right? We've already dealt with these. We know that they're secreting the hormones ADH and oxytocin, right? So that's what we've been talking about. But how do they actually get into the bloodstream? Well, that's where this hypophyseal portal system comes into play. And what that is it's a set of blood vessels that connects the hypothalamus to the pituitary gland. That's the hypophyseal portal system. But if you notice, there's 2 components. There's a superior artery, and then there's an inferior artery. And you really need to distinguish between those 2. Again, we're expanding on the details that are expected to know for this upcoming lecture exam here. All right, obviously, we still have a ways to go with that lecture exam. We still have to get through chapter 22, the immune system, but that's what we're moving towards. All right, and so I'm going to introduce you to a unique concept of capillaries, and we're going to really get into capillaries in Unit 2, but I want you to know that capillaries, there's 3 different types, and I want you to know what fenestrated capillaries are. We'll build off of this later, like I said, in Unit 2, but these are capillaries that have pores in them, and if you didn't know what capillaries are exchange blood vessels. Okay, and so these are fenestrated capillaries. These are large pored capillaries that allow for bigger exchange because, of course, we're dealing with peptide hormones. Normal capillaries, which are called continuous, which we'll get to later, they really allow the exchange for only very basic things like individual amino acids, individual glucose, where fenestrated is going to allow for a lot more. And so that's important to note that the hypophyseal portal system consists of fenestrated capillaries. And this is what's going to allow the hormones to be released by the hypothalamic neurons and to actually enter into the blood, or to be released. It doesn't even need to be the hypothalamic neurons. It can just be hormones coming out of the hypothalamus, those regulating hormones as well. All right, so regulatory hormones, they're going to travel using the superior hypophyseal portal system. So HPS stands for hypophyseal portal system. As you see, I've abbreviated it there. And that's what's going to stimulate the anterior pituitary. So that's really the bullet point you need to know is right there. The superior hypophyseal portal system is what allows regulatory hormones that are secreted by the hypothalamus to travel down to the anterior pituitary. All right, as far as the inferior hypophyseal portal system, and this was mentioned in the recent lecture, the part 2 portion of lecture 2, this is what carries the hormones this carries the hormones of the posterior pituitary into circulation because, again, remember, it's only secretion that occurs at the posterior pituitary. There's no production. So notice here's the supraoptic, here's the paraventricular nuclei, and as you can see, both of those neurotransmitters end at the inferior hypophyseal portal system, and that's why these are actually hormones and not neurotransmitters. They're hormones because they're in the bloodstream. They are not neurotransmitters because they're not communicating with other neural tissue here. So because it's in the blood, it makes it a hormone. All right, and so overall, the hypophyseal portal system, really important for quick regulation of hormones, and it's really the communication route between your master endocrine gland, being the hypothalamus up here, down to your pituitary gland sitting below. So really simple concepts here, very easy for me to either test on the superior or inferior. Okay, that's one perspective, or just talking about what is the hypophyseal portal system, and really as long as you know, generally speaking, the hypophyseal portal system connects the hypothalamus to the pituitary gland. You have the main point, and then you have to incorporate the detail that it's fenestrated capillaries that make this up. So all the blood vessels you see down here are truly supposed to represent mostly capillaries that are fenestrated. All right, so know these are straightforward, quick bullet points that I can easily ask about. There's really no big picture beyond just knowing the details here. Hypophyseal portal system, superior versus inferior, and do you know fenestrated capillaries? So don't overthink this. Definitely review it, because they should be the simple points that you're able to get, because you don't have to really go beyond this PowerPoint here, this PowerPoint slide. All right, so the histology slide. So this is the histology image. I do often have lab lectures where I just focus on laboratory concepts. Hands down, this is a lab concept. But, it's fair game for me to, utilize images that look very similar to this on a lecture quiz. So you might see an image like this, and it might be as simple as, what does this image represent? And as you're going to see, especially if you listen to the lab lectures are a little more easier to follow, because literally I'm doing all lab concepts, so you can see how different the histology is. And there is no histology, no other endocrine histology image that looks two-faced like this. If you notice, we have a dark face, and then we have a smaller, lighter face. We have a smaller, lighter face, and a larger, darker face. Here, smaller, lighter face, and a larger, darker face. So don't get caught up on left versus right, because that's not what this is. Really, really need to be paying attention to the dark side versus the light side, because the light side, as you can see, is the pars nervosa. Okay, and that's the same reigns true if you look at the models that we have. There's a lighter side on the model and the darker side. And again, it's the lighter side being nervosa because it's neural tissue mostly. And so when we break this down histologically, we have 4 unique histological layers. Out of those 4, I really expect you to be able to recognize pars nervosa being the lighter region. Okay, hands down. Lighter region, it's going to be pars nervosa. Notice pars nervosa is on the right here, it's on the left here, it's on the left here. What I would encourage you to do if you want to get practice at this, I would use Google and I would just type in pituitary gland histology and look at ones that look very similar to this. That's what you should be doing to making sure you're kind of understanding the concept. And then, of course, when we're in class and during lab, you certainly can use the histology slides to make sure in the microscopes that you're able to identify these components. So typically what I would ask is, if I had just this single image up there, obviously these labels would be removed. But often what I would ask is, what is this a histological slide image of? Well, in this case, you would just put pituitary gland. And then maybe I put number 1 in here, and I say, what is this region? If you notice, the term you need to use is pars distalis. I expect you to be using what I consider the histological terms, because that's the perspective we're looking at. So here, hands down, the correct answer is pars distalis. Here, if I had number 2 here and I said what region does number 2 represent, you would say pars nervosa, not the posterior lobe. Usually I give half credit on a quiz, and on the lab exams I usually give you no credit for those answers. It's histology, perspective matters, my choice, and again, this is me as an instructor. Your other instructor, if you're taking a different course, your other instructor might be okay with you putting anterior lobe. I am not. I'm expecting pars distalis here, and I'm expecting pars nervosa. Pars nervosa here, pars distalis here. Pars nervosa here, pars distalis here. Now, if we have really nice images with this kind of middle region here, it's okay for me to label. If you notice, this color right here is slightly off, and that's going to be pars intermedia. That's fair game. Often if I do it, I usually do it as extra credit for students, but this little region right there, pars intermedia. Okay? That's it. I do not do pars tuberalis, so you do not need to get caught up on pars tuberalis. I don't label it. I don't expect students to know it. In fact, I pointed this out. If I point to kind of a stock thing on a model, because again, a model is not the same as histology, it would not be pars tuberalis. It will be that infundibulum. So make sure you know what connects what connects the hypothalamus to the pituitary gland. That was the infundibulum. You need to know those and be able to recognize it. But again, histology, pars nervosa, pars distalis, pars intermedia. This is a really good pars intermedia. This is an okay one. You can see it's on the nervosa side, so that's going to be pars intermedia. But as I continue to stress, usually I just save that for extra credit. Now the final question, so can you identify this as the pituitary gland as a whole? Can you identify pars distalis, pars nervosa, and pars intermedia if it exists? This one I probably wouldn't use. I don't like it, but this one's really good. This would probably be the histology slide I would use for it because it's really clear. And then the final thing is if I ask, name a hormone secreted from this region. Often when it's pars distalis, I'm usually asking you to name 2, maybe 3 hormones. When it's pars nervosa, I'm usually asking you to name 1. Obviously with pars nervosa, ADH, oxytocin work. With pars distalis, you have 6 to choose from, so just choose wisely. It's pretty much all the anterior pituitary hormones. So that's the histology. If you're like, I'm still confused by this, I would encourage you to listen to the lab lecture where I really break these down in full details, but I'm going to move ahead because that's about as much time as I would spend in a true lecture. All right, but hopefully you're starting to see that there was a high degree of regulation. And so figure 18.18, I believe that's this top one, really shows you the hypothalamic regulation. And it includes the hormones I expect you to know. Notice TRH, CRH, and GnRH. I told you need to know those. You should know what they stimulate. So you're getting a lot of figures here. You're getting concepts that are building off of each other. I hope you're spending time with these figures. You're going to hear me say this over and over this semester, that these figures will do you more justice than just listening to lecture. You need to spend time with figures. If you can explain the figure, you probably have a really good understanding. Now, when we get to class, our first class, one of the first activities we do is talking about this, essentially the negative feedback loop. If you notice, so here's the releasing hormone. It targets the anterior lobe. That stimulates the anterior lobe to release prolactin. And now notice prolactin, which targets the mammary glands to stimulate milk production, but notice it also goes back and it causes inhibition. So that's the feedback loop. And notice it's stimulating the inhibiting hormone. You don't need to get caught up on that. That's what this is showing you. This is an inhibiting hormone, which is going to slow down production. The main point is, notice it's a feedback loop. Same thing if we go to growth hormone. Notice there's the growth releasing hormone that causes GH to be released. That goes to the liver. That produces somatomedins. And then notice we go back and we block the releasing hormone and we stimulate the inhibiting hormone. It's just showing you that typically when hormones are released, their secondary effect is to slow down the secretion of that specific hormone. Okay. Hopefully the activity we do in class and then that becomes homework, hopefully that starts to make sense of this feedback loop. These are a little bit more complex examples because they're including the inhibiting hormones and releasing hormones. I don't do anything with inhibiting hormones, as I've said. All right, and then here, this figure at this point should start to make a lot more sense. And hopefully you're getting the concept of somatomedins being important for growth hormone to actually function. So somatomedins is related to growth hormones. But at some point when you look at this figure needs to make complete sense here. ADH, do you know what it does? Do you know what oxytocin does? All right, LH, FSH, prolactin, growth hormone, thyroid stimulating hormone, and adrenocorticotropic hormone. All those hormones were discussed at this point. This figure should start making more and more sense. I would encourage you to review it, spend a lot of time here. Notice here's the indirect control through release of regulatory hormones. It's an amazing figure. You need to start saying, yeah, these things are making sense. Some of them should be making quite a bit of sense right now. They might not make perfect sense yet, but again, revisit this figure until the point that you're like, yeah, I know ACTH targets the adrenal cortex to cause the release of course, our glucocorticoids or cortisol and cortisone. That's what I've stressed. Notice they're showing you the direct nervous system control. This is sympathetic control of the adrenal medulla causing release of epinephrine, norepinephrine. At this point, you should know these are the catecholamines and the tyrosine derivative hormones, right? And they're water soluble. So a lot to be starting to work through. And if that didn't make sense, that's why I'm spending a lot of time on these figures, is I'm showing you, you need to be building off of content that we previously covered and continue to move forward and then reflect on this again. Notice direct hormone release, sensory stimulation versus osmoreceptor. So notice osmoreceptor ADH release, sensory stimulation, oxytocin. You need to make sense of those things. Now, where we're heading with lectures, we're going to start looking at thyroid hormone. We're going to start looking at the adrenal cortex. We're going to be spending time where we're going to get into inappropriate hormone secretions and what happens. Like, if the thyroid hormone is secreted too much or too little, we have terms for that. So we're going to start with growth hormone because I think it's pretty easy to see. We know what growth hormone's effects are. They stimulate growth of cells. Right? So too much growth hormone during childhood, what would that lead to? And that's what these figures here, of course, are showing you. These are individuals that have growth hormone being secreted excessively during childhood, and that, of course, leads to gigantism. These are really simple questions. Anytime I mention, kind of disruptions to normal homeostasis or disruptions to a normal system, I tend to test on those. So if I talk about, like, when we get to the lungs, I'll talk about pneumonia. You're going to want to know what that specifically causes. So gigantism, something to know. Too little growth hormone being secreted, you should know that leads to, of course, dwarfism. Often this is, genetic. We're pretty familiar with individuals that have dwarfism. They have a lot of the common characteristics associated with dwarfism. But fortunately, this understanding that it's related to a secretion, too little of secretion, or a hyposecretion, meaning a low secretion, there's ways to counter this by giving supplements of growth hormone. So hopefully these things like dwarfism start to really become a thing of the past, and they certainly are becoming less and less prevalent and having better treatments. And then you can have high levels of growth hormone being secreted during adulthood. Obviously, they're showing you this sort of Shrek-like individual. Literally, we're talking about having like a big bone disease. It's not really a disease, but it's having big bones. And that would be acromegalia, which is over here. This individual, their bones, because of course your bones can no longer lengthen. So at some point, bone lengthening stops, and what happens is the bones continue to grow in width. And so that's what acromegalia is somebody having literally big bones. Final thing to note, which I mentioned already, is you should know growth hormone will eventually lead to the production of something called somatomedins by the liver. So at the liver, growth hormone gets converted to somatomedins. And these are called insulin-like growth factors, i.e. they contribute to growth as well. So if we go back to the figure over here, you're seeing growth hormone hitting the liver, becoming somatomedins, and that stimulates bone growth as well. Or if we look back here, you can see growth hormone. Notice epithelia, adipose tissue, liver, i.e. stimulating growth, and then somatomedins, stimulating growth of skeletal muscle, cartilage, and other factors. So in this case, growth hormone really becomes somatomedins to have their biggest impact. But generically speaking, if I mention growth hormone and I don't have somatomedins on there, you should know that it is, in fact, growth hormone that stimulates the growth of other organs, okay? Somatomedins is the more direct hormone or the more direct chemical that gets changed and actually causes those impacts. I am not that big of a stickler on that. I don't expect students to know, hey, somatomedins is the more correct answer. You would either see growth hormone as an answer choice or somatomedins, not both. So if I want you to know which of the following hormones stimulates growth of tissues, it'll be one or the other, not both. All right. If you have another instructor who spends time with growth hormones versus somatomedins, then you better know the differences between them. For me, I keep it pretty generic. All right. So that finishes up our second lecture here, and this is the entire group note review. You should be able to answer these bottom four after having listened to specifically this part, but obviously we're through all three parts, therefore every single one of these questions technically you should be able to answer at this time. This, I haven't really stressed it as much as I will be, but if you can answer all of these and feel pretty comfortable with it, this is a great way to self-reflect on where do you stand with the content. Let's just say you can answer, 5 of the 8. Well, you've understood in lecture 2, you've understood about 50 to 60% of the concepts because you're missing about 50% at that point. All right, that's how to use these. Make sure if you can come up with great answers to these on your own without looking at notes, you are well prepared, and that's how I would be using my basic group note reviews, is double checking how prepared you are. All right, I hope the lectures are really starting to make sense. This was technically the 5th lecture or 5th episode, if you want to call it that. Obviously, this is technically lecture 2, part 3. I did lecture 1 and I had 2 parts, so with this being essentially this is the 5th episode so far. All right, I hope things are going smoothly. I hope you all appreciate these and are getting a good amount of knowledge from them. If you have questions, please bring them to class. Certainly we can work these out. Most of our class time, especially if this is a hybrid course, most of our class time is spent on the lab stuff. So if you have specific questions, bring them to class because I don't cover a lot of this during our lab time, because it truly is supposed to be preserved for lab time, not lecture. All right. Take care, everyone, and look out for part 3. When we move on to part 3, we will really start breaking down the thyroid gland. We'll break down the adrenal gland as well. And then we're kind of on to the final concept, which is, again, what we've been building towards, which is how water-soluble and lipid-soluble hormones function. All right, but we'll be covering that soon. Take care, everyone, and I look forward to seeing you at the next lecture.